So this is an outline of the talk. I'm going to go over where we are in exposomics, as well as the definition of exposomics, the role of philosophy in defining exposomics. I'm going to use a lot of metaphor in this talk, so it's going to be a little bit different in that regard. But I'm going to talk about how, as an analogy, geography was actually important in genomics as well, so that's why it may be important in exposomics, and why a human exposome map really needs a purpose and how we can actually learn some lessons from how genomics constructed its map. And once we take those lessons, perhaps we can actually create a human exposome map that can be used for research as a research infrastructure tool. So, exposomics is really about exposure and measuring exposure, or exposure science. It's really exposure science on steroids. The advancements that are being made in exposomics are primarily in measurement technology. And it's really roughly where genomics was about 15 to 20 years ago. Just as genomics learned about the genome's function and structure from mapping the human genome, we can really accelerate our understanding of the exposome if we map it. And innovation and rigor in genomics really didn't precede the rise of the Human Genome Project or the Human Genome Map. It actually followed it. So most of the discoveries were made afterwards. So an exposome map, I'm going to argue, is going to actually innovate a lot of research in exposomics in general, and help us understand health disparities in particular, because the reasons why health disparities exist are not due to genomics, they're due to differences in environment, defined as the exposome. So not just the chemical environment or the physical environment, but also the social environment, the infectious environment, all the different environments you see on this slide. So I like to read. I'm a big fan of Latin American literature, particularly mystical realism. And there is a particular author I'm very fond of, Jorge Borges. And he wrote a short story about a man with a perfect memory, Aurelio Funes. And it was called Funes the Memorias. And in this story, this man with a perfect memory who could remember everything, everywhere, all at once, all the time, and it slowly drove him crazy. And these are some excerpts from the story. So Funes remembered not only every leaf of every tree in every patch of forest, but every time he had perceived or imagined that leaf. It irritated him that the dog of 3:14 in the afternoon, seen in profile, should be indicated by the same noun as the dog of 3:15 seen frontally. To him, each memory of the dog was unique, not just the dog itself. So everything was cataloged in his mind, everything he experienced. In essence, he had no insights on life. He could only recite facts in minute detail. In the story, Borges talks about how for him to describe yesterday would take more than 24 hours because he had to describe everything that happened, and the sentences would take longer than the experience. And so he could never describe anything. So where does that get us into philosophy? So I'm going to talk about the difference between realism and nominalism. So realism is a philosophy in which the world is defined by particulars and universals. Particulars share characteristics of universals. For example, Cody is a dog, Frodo is a dog. Frody and Kodo in realism are particulars, and dog is a universal. In nominalism, there are no universals. There's only particulars. For example, there is no good or evil. Each behavior and action is unique. Cody is Cody, Frodo is Frodo, and there is no dog. Taken to an extreme, Cody this morning is different from Cody who is playing with Frodo now. So what Borges was describing in Funes' Memorial was nominalism, where every event is unique, and how remembering everything and all this information that he was collecting actually was mostly useless and was inhibiting his ability to live life. So how does that relate to the exposome? So I'm going to argue that exposomics is in danger of becoming a nominalistic science. So the exposome is composed of every exposure to which an individual is subjected from conception to death. That's what Christopher Wilde defined as the exposome in 2005. In 2012, he expanded on it a little bit. He talked about internal processes such as metabolism and hormones and microbiome and inflammation, and external processes of radiation and air pollution, social, economic, and psychological influences, as well as exposure timing was added. So whether or not you're exposed when you're an infant or you're exposed later on in life may change the dose-response curve. Well, if we're trying to measure all these things and factor in all these things, we're really in danger of becoming anomalous measurement science by trying to measure, again, everything, everywhere, all at once, including all the time. So how do we redefine exposomics towards realism? Well, instead of measuring everything, maybe we should think about trying to model it or impute it. We don't necessarily have to measure everything. We can measure the relevant environment by defining its correlation structure and its interrelationships. Measure just what we need and model the rest, in other words, or imputation. There are likely rigorous, realistic shortcuts. We can sample weekdays and weekends for people. We don't have to sample every minute of every day. We can use models to fill in the gaps. And nowadays, with AI and machine learning, we can probably model and impute at a much greater level than we could have even 10 years ago. And there's a lot of things we can measure now that we know are important. They've been scientifically vetted. A lot of them have been discussed at this meeting, but we don't have a map for them. We don't have a universal map, and that's really what I'm advocating for, is a universal map. We have data on chemical, physical, social, dietary, and behavioral exposures. We can assess the relationship to important contextual factors. We can use geographical positioning devices to also understand the role of place, and place is very, very important. But we can also attach SES, socioeconomic status, age, neighborhood characteristics, and occupation and such to all of those spaces. And once we understand those spaces, we can start to understand, as Dr. Bullard said, why is place so important? Where you live is a better predictor of your future mortality than anything in your DNA or anything we can measure in your DNA. And so we need to really invest in understanding place, and place is just a map. And if we do this, if we create an exposome map, we're going to have a research tool that's going to enable real exposomic research, and I'll try to explain how that's going to work. Well, first we're going to apply lessons from genomics to exposomics. So the genome is a map. Geneticists even refer to it as a map. So how can we facilitate exposome-wide association studies using an exposome map, just like genomics uses the genome map? Well, if you think about it, chromosomes are the landscape of the genome. We use them to define place for genes. Early maps were two-dimensional, kind of rudimentary. We didn't factor in the three-dimensional structure of DNA, but we knew that certain genes that were close to other genes on the same chromosome tended to be inherited together. So as genomics evolved, we started to realize that areas that looked really far apart on a simple map were actually close together, because in reality there's a three-dimensional structure, and DNA wasn't a line, it wasn't a string, it was a crumbled up ball. Crumbled ball of string, so that areas that we didn't realize were next to each other actually are next to each other. And as we start to create this more three-dimensional map, we have a better understanding about how genes function. So genomics also leveraged this geography to create what's called a haplotype map, or a hap map for short. This was in the early days of genomics. And in fact, hap maps are how genome-wide association studies actually came to be. The genome map showed that variants that were nearer each other tended to be inherited together. And these regions were known as haplotypes. So a whole group of variants right next to each other tended to go together. So if you had this variant, you're most likely to have that variant, because there's a really strong correlation between them. So essentially what the hap map did is it took advantage of how single nucleotide polymorphisms, or SNPs, and other variants were organized geographically on chromosomes in order to conduct genome-wide association studies. Essentially, this was exploiting geographic correlation in the genome landscape to accelerate research. So HapMap is actually used in GWAS studies, or genome-wide association studies, by using what are called haplotype tag SNPs. These are single nucleotide polymorphism SNPs that represent a group of highly correlated SNPs. So if you measure one SNP, you know a whole bunch of other SNPs. Hundreds to thousands of other SNPs tend to be inherited at the same time. So you don't have to measure all those SNPs. You just have to measure these tag SNPs. An association between a tag SNP and a health outcome represented an association with a geographic region on a chromosome. So the question then, if there's an analogy here, is that are there exposotype tag SNPs that we can use similarly in exposome-wide association studies? So if there's a genotype, maybe we also have an exposotype. like all the environmental exposures that we're exposed to that we occur in our daily life and that have occurred in our past would be our exposotype. And maybe there are particular ones that tag other ones. And if chromosomes are the landscape of genes, then if we're going to discover these exposotypes, then Earth is really the landscape of the exposome. So I think by assessing the exposome's correlation structure, we can conduct really good exposome-wide association studies. So how would this work? So a typical genomics study happens in multiple stages. So there's what's called a discovery stage, where you have people with the disease in a control group, and you look at associations and you measure hundreds of thousands of SNPs. Most of them tag SNPs for the haplotype tag SNPs. And then you replicate it in a subsequent population, and then you zero in on what you think are the functional variants, and then you do functional studies afterwards. Well, we could probably do this in exposomics if we understood the correlation structure of the exposome. So if we measured millions of people like they have in genomics, and we measured their environments, and we looked at what are their exposotypes, and we looked at the correlation structure of those exposotypes, and we associated that with addresses and age and life stage, we could actually create an exposome map that would tell us what these exposotype tags are. And using those tags, we could efficiently do exposome-wide association studies. So if the geospatial map of everything, we can determine the correlation structure of the exposome. And then we don't have to measure the exposome anymore. We don't have to be Ireneo Funes, who's slowly going crazy because we're trying to measure everything. We can start to impute it. An imputation would just be filling in the missing variables. So if red are missing data points and green are measured data points, you can impute the red data points if you know the green data points, if you know the correlation structure of all these different variables. So with machine learning and artificial intelligence, I think this is actually quite possible. So I'm going to call these exposotype tag measures, and that this is a strategy for exposome-wide association studies. So these are measures that tag a higher probability of other exposures being present. So if you measure the tag measurement, you don't have to measure the other ones because you can infer by correlation that the other ones are likely present. An exposome map will identify what exposures are associated with tag measures, such as the occupational exposome or the neighborhood exposome. So you don't have to measure everything in your neighborhood if you think about it. You just have to measure certain things in your neighborhood because there's a correlation structure because of the geographical proximity of different things in your neighborhood. In exposome-wide association studies, exposotype tag measures might let you zero in on the most likely relevant exposures like haplotype tab 6 in genomics, and you can deep sequence people's exposomes to find the specific external environment, internal biomarkers, social determinant of health that is correlated with the tag measures in sort of these multi-stage studies that genomics does. So these are some examples that I think will be exposotype tag measures. So they could be home, location, so where you live matters. We know place matters. So if we know your address, that tells us a lot about you. And we can start to do health studies, and we can start to zero in if we find that particular locations are more likely to be associated with poorer health outcomes. We can start to do the equivalent of deep sequencing in those neighborhoods to try to figure out what it is that's actually causing it. And this is really about discovery research. And it's actually, in some ways, very identical to Dr. Bullard's work, because he was discovering things that were unknown scientific or even unknown within a community. A lot of our environment actually is unknown, and an exposome map would actually allow us to start to discover those things. Other things that will tag other types of measurements are life stage. Infants have certain types of diets. Children have certain types of behavior. They're more likely to get exposed to things like lead. Lead poisoning actually peaks at 2 years of age because kids have a lot of hand-to-mouth activity at that age. It's completely normal. If there's lead in their environment, lead in the dust, they get lead poison. If there's no lead in the environment, they don't get lead poison. But all kids who are 2 years of age do that. But that behavior disappears later on in life. So life stage matters. So there's an exposome life stage correlation. So certain exposures occur at some ages, but not at other ages. Income is certainly going to tag a whole bunch of different types of exposures, such as pollution or nutrition or educational opportunity. And products are going to tag their ingredients. Occupation is going to tag different types of exposures. And there's probably hundreds of thousands of different exposotype tags. But the idea behind this is to try to do discovery research, because we don't really understand the role that environment plays in our lives because we haven't made the investment yet. So this type of map actually should be able to facilitate that type of discovery research. So it would look something like this. You would do a study where you do your measurement of thousands of exposotype tags in one population. You try to replicate it because probability is that most of those hits are going to be false positives. That's what genomics kind of discovered the hard way. And once you replicate what is actually causing the effects, eventually you go through multiple stages where you actually start doing some risk prediction, and then you can start doing interventions. And this is much faster than doing a hypothesis test of one at a time because hypothesis tests take a long time. So by interrogating the exposome, we can zero in on the factors that are mattered in a matter of a few years as opposed to decades. So other benefits of the human exposome map. First, we can use it the way genomics uses haplotypes for exposome-wide association studies, but we can also use the correlation structure of the map to impute environmental exposures. We don't have to measure everything. We can start to use AI, artificial intelligence, and machine learning to try to figure out what exposures you're exposed to if you know a certain subset of exposures. Plus, if the maps go back in time, we can also go back in time. So there have been maps that have been correlated over many years. Dr. Bullard made a lot of really useful maps that tell us a lot about the geography of the environment, and that tells us about past exposures. And we know the science is very clear that if you're exposed to something as a fetus, when your mother is pregnant with you, or if you're exposed early in life, it's much more toxic than later in life. So we actually need maps that go back in time, because while we want to protect our children that are being born now, the effects of things that happened, in my case, in the 1960s, are actually why I have certain health problems or are healthy today. So we have to go back in time if we want to figure out what's happening to everybody in this room. We can start today for our children and our grandchildren, but for us, we have to go back in time to understand what's happening. Today, there's actually a lot of genomics research going on that actually has developed pipelines to try to impute what genomic variants people have by doing genome-wide association studies or sequencing. So there's been a lot of investment in that. One day, I think we should be working towards being able to do that for the exposome, so that when people measure an exposome on a group of people and you know where they live and you know what their occupations are, and you know their age, we can start to impute the neighborhood values, and those neighborhood values can help everybody in that area. So we actually can do a lot of this work modeling what genomics currently does. Other benefits, it's going to, as I said, enable exposome-wide association studies so we can discover not just what environmental factors are causing our health problems, but where they are, because place matters. And a map actually tells you where things are, and that's going to be very, very important for health. So even things like lead poisoning, we don't actually know where all the lead is. So Flint, Michigan didn't know where all the lead was. But this type of map could actually have pinpointed where the problems were if we had this type of map. It'll enable gene-environment interaction research, so the geneticists, I think, will probably get on board, because genetics only explains a small proportion of why we get ill. Genomics is limited without exposomics. And I think most importantly, it's going to enable health disparities research on a grand scale, because we're going to start to discover where these environmental factors are. And I think the maps are going to illustrate why health disparities tend to correlate with place. And it also is, we can go back in time and enable developmental origins of health and disease research. And this isn't really reinventing anything or inventing anything, it's really reinventing anything. So we actually are very good at making maps. Dr. Bullard showed several nationwide maps. This isn't rocket science, this is something we can do. What we're really talking about is bringing all these maps together into one place so we can start to look at how they overlap. And if we start to look at how they overlap, we can see how much lead poisoning overlaps with air pollution or overlaps with healthy built environments. What we need is a resource where all these things are in one place, which is going to take a significant amount of effort. But you can map happiness. So this is, I think this is Itai Klug's paper, actually looking at spatial clusters of happiness using data from Twitter, now called X. So using natural language processing, which is a type of machine learning, he looked at happy tweets versus non-happy tweets and actually mapped the geospatial cluster of them. And this is something that Dr. Wright, Rosalind Wright showed earlier today about how redlining correlated with air pollution. So neighborhoods that are redlined, which were appropriately colored red, actually have higher levels of air pollution than neighborhoods that weren't. So what this map is going to show is that intersection between race, ethnicity, and pollution, and poor disinvestment in environments. That's why we need maps, because maps actually show why place is so important. And then this is Massachusetts Neighborhood Sentiment and Safety Index, which is actually derived from public databases, but just to show that there's ways to map the social environment, there's ways to map the physical, the chemical, even the infectious environment. There's lots of COVID maps that were made during the pandemic as well. So you can map anything. You can even map biomarkers. If you're mapping metabolomic data, there should be a way to put it on a map. So summary of an exposome map. Well, as I said, everything can be mapped. Biomarkers can be mapped. Climate, air pollution, green space, noise, socioeconomic status, social media, buildings, green space businesses. There's real estate databases that can be mapped. Health data can be mapped, such as depression, cancers, and infections. So you can overlay where the cancers are occurring when they occur in a geospatial cluster with what the environment is in that cluster. And there's nothing really stopping us from doing this rather than the will to do it, because all of these data exist, they just exist in silos. So what we're really saying is we need to start the hard work of bringing all this work, all these maps together. Because maps are going to accelerate our ability to discover what the exposome is doing to our bodies. And I think the time is right, because the computational infrastructure to do this with imputation and artificial intelligence is here now. And if we create maps that go back in time, we actually may be able to start to understand some of the health problems that we're having today as adults, because many of them had origins in our childhood. And so I read a lot of science fiction, so this is always one of my favorite quotes. I didn't understand it when I first read it. William Gibson wrote a novel called Neuromancer. Neuromancer was the inspiration for the Matrix movies. And this is a quote from Neuromancer: "The future is already here, it's just not very evenly distributed." And I think what he's saying is there's people that have and people that don't. And so the people that have owned the future. We just have to make it more evenly distributed. And that's really what environmental justice is about. And also, I think NIH data sharing may be a vehicle to help build a map. And not all analyses using the expo map need to be ecological. There's ways to do this with the All of Us program or the ECHO program, where we can actually map the biomarkers that are collected. And there's many other NIH large cohorts, multi-ethnic cohort, Women's Health Initiative. All of them have biomarkers that can be added to the map. And biomarkers may actually identify unknown toxic waste exposures. So if we have maps of biomarkers and we know people in a particular geographical cluster are exposed to something, there's probably something in their environment that's doing it. And so it may actually lead to more discovery at the place level. And as I said, we don't have to reinvent the wheel. We just need a program to integrate all of these different maps and come up with a complete exposome map for the United States. And hopefully one day the globe. And we can use it to define the exposome. So I've been in this field for a long time, so I've worked with a lot of people. Their names are on this list, including Daryl and Paul, so thank you, and Itai, who's also in this room. But a lot of this work came out of that, and was actually inspired by the public health exposome that Paul and Daryl put together. So thank you. That's my last slide.